Good evening, everyone, and welcome to this new episode of Red Talk. My name is Alexis Talbot, and together with Jaime Fuentealba, we will have the pleasure of moderating this session. Thank you all for joining us today. We are very delighted for what promises to be a truly exciting discussion. First, I would like to warmly thank the Carnot Kalim Institute and the entire Kalim team for organizing this Red Talk. So as you all know, CAR-T therapy has transformed the treatment paradigm in hematology and especially in malignancies, and has become one of the most remarkable achievements in translational medicine. So today's episode will explore the evolving landscape of CAR-T therapy, from ex vivo approaches to emerging in vivo CAR strategies and their impact on current and future therapeutic development. Before we begin, a quick reminder. Please feel free to use the chat and Q&A box throughout the session to share your comments and questions. So please do it, engage yourself, ask questions. Jaime and I will be very happy to bring them into the discussion later. For today's session, we are truly honored to welcome Professor Michel Sadelain and Dr. Justin Eyquem. The good news is they are very famous and well-known, so I will be very brief. Professor Michel Sadelain is internationally recognized as one of the founding pioneers of CAR-T cells and a founding figure in the development of CAR-T design. His work at MSKCC previously and now at Columbia University has been instrumental in bringing CAR-T cells to the clinical reality now. Dr. Justin Eyquem is a leading expert in CAR-T cells engineering, known for his innovative work on precise CAR integration and advanced synthetic engineering approaches for the next generation CAR design to improve efficacy, safety, and persistence. So together, Michel and Justin will offer a complementary strategic perspective from ex vivo to in vivo CAR approaches. Thank you again, Michel and Justin. It's truly a privilege and honor to have you here for 1 hour just for us. Before moving to our speakers, we will first hear a short introduction to today's topic by Jaime. Jaime, the floor is yours.
Thank you, Alexis. So, well, as you all know, 10 years ago, a patient with certain relapsed or refractory blood cancers had extremely limited options. And today, a single infusion of CAR-T cells can induce complete remissions in more than 80% of patients with acute lymphoblastic leukemia, and between 38 to 63% in large B-cell lymphoma. 7 FDA-approved products are now available, spanning B-ALL, several lymphoma subtypes, and multiple myeloma. And over 30,000 patients have been treated globally. This is clearly a turning point in oncology. However, this revolution is clearly unfinished. Even in hematology, responses are not universal, and relapses unfortunately occur, driven primarily by poor T-cell persistence and antigen escape. And in solid tumors, CAR-T cells face completely different challenges, overcoming tumor target heterogeneity, poor infiltration, and they have difficulties in navigating a highly immunosuppressive tumor microenvironment. We are seeing some promising signals recently. However, of course, there is no breakthrough yet, nothing that matches what we already saw in the past in hematology. And even where CAR-T cells work, most patients worldwide cannot access it. Manufacturing takes weeks and costs hundreds of thousands of dollars. So today, these are the 2 main questions that our speakers today are going to tackle. So can we engineer better, more durable CAR-T cells capable of fighting solid tumors? And then can we generate CAR-T cells directly inside the patient, eliminating manufacturing completely? So please, Michel, the floor is yours. We're really looking forward to your presentation.
Thank you, Jaime. Thank you, Alexis, for the very kind introduction. Well, I will start with going over the origins and evolution of these molecules that we named CARs over 20 years ago. And going over a little bit of the history, I only want to explain where we stand today and why we have learned what we have learned, and why the obstacles we face today are what they are. Vertebrates have the amazing ability to generate very vast numbers of antigen-specific receptors through the recombination of genes. And they do this in two different cell types, generating two types of receptors. B cells express a B cell receptor, which they can secrete in the form of an immunoglobulin. And these circulating antibodies surveil extracellular fluids and can also engage the surface of cells and particles. T cells, on the other hand, generate a receptor that sees inside cells. To see inside the cell, peptides have to be brought to the cell surface, and they are presented by HLA molecules, which are engaged by these T cell receptors. So B cells contribute to surveil extracellular milieux, and T cells see inside cells. CAR T cells represent really a reallocation of these properties and generate essentially an unnatural new cell type. Like T cells, they engage directly with their targets, but they do so independently of HLA and do not require antigen processing by the cells that they engage. Furthermore, these CAR molecules that are introduced into T cells not only target a T cell, but they reprogram cellular functions. So I'll just quickly walk you through how these molecules came to be before discussing current challenges. I'll go very quickly and say that if we go back 3 decades, I think the two pillars really that opened up this field were, on the one hand, the ability to introduce genes in primary T cells. And that was fundamental because these molecules could then be assessed not just in T cell leukemias, as had been done until then in the field of immunology, but in real cells, that need to see antigen to proliferate, that undergo activation-induced cell death, that depend on co-stimulation, etc., things that T cell leukemias don't depend on. And then, on the other hand, some receptors, new receptors, came to be in the early 90s, following the cloning of the CD3 zeta chain of the CD3 complex of T cells, which led to eventually the recognition that you needed to merge different signaling domains, as shown here in the upper right of this slide, to achieve a molecule that could not only retarget a T cell through an scFv to any cell surface antigen independently of HLA, but also sustain its functions and proliferation upon repeated encounter with antigen. And those are the molecules that in 2003 we named CARs. What I really want to introduce next is this prototypic target that we selected actually in the mid-90s as one that we would explore in our animal models and one that we thought we would use someday in the clinical setting, and that is CD19. We thought we would experiment in the realm of hematological malignancies for a number of reasons, but in particular because we knew from the graft versus leukemia responses that donor T cells in the context of a transplant could efficiently migrate to bone marrow and lymph nodes in particular. And there was, of course, an unmet medical need back in the day and still today for these B-cell malignancies. We chose CD19 over what appeared to some to be the more obvious choice of CD20 because of its abundance, its earlier expression in the B-cell lineage, and apparently homogeneous expression in a number of B-cell malignancies. CD20 was already famous in the 90s. It's the target of the antibody rituximab, which is still used today. But CD19 was more abundant at the cell surface, and we thought that might be important, which turns out to be the case, where today the sensitivity of these receptors of CAR receptors has become a very significant question. I'll just mention there were other reasons for choosing B-cell malignancies and CD19, and one of them was to ablate normal B cells, which we thought would circumvent the potential immune response, antibody response of the host against the CAR molecule. So anyway, in 2003, this paper mentioned in the lower left came out and showed that you could use the recombinant retrovirus harboring a synthetic gene that coded for a CAR molecule specific to CD19, and that a single infusion of these T cells in mice could induce long-term remissions and even cures. And from there on, it was up to cell manufacturing to bring this to the clinic, which at my center, Memorial Sloan Kettering, was beautifully accomplished by my colleague, Isabelle Rivière. So many of you, we need not review the earlier clinical data, but three groups, the NCI to whom we had given our molecule, the the Children's Hospital at Penn, CHOP, and our team at MSK produced these very remarkable results in patients with refractory relapsed B-cell malignancies that soon led to more trials confirming these results and ultimately the approval of these medicines. And it's in discussing this that I refer to these molecules as, to CAR T cells as living drugs. So these were the first two CARs to be approved by the US FDA back in 2017. They're both specific to CD19, and they both comprise these dual signaling entities, one for activation and one to amplify these the signals. It's quite remarkable that both these original CARs, if you like, one using the costimulatory domain of CD28 and the other one of 4-1BB, produce such remarkable disease responses in leukemias and also in lymphomas. Remarkable given the differences in signaling that they actually afford. And although that's not our topic for today, it's remarkable how much we continue to learn from comparing these two cousins, if you like. So there are, as we just heard from Jaime, 7 CAR T cells that are approved in the US and in Europe today. 5 of those target CD19, 2 of them B-cell maturation antigen, a key antigen in multiple myeloma, and some use these 28-based CARs, and some use the 4-1BB-based CARs. I'll just mention briefly sort of a second tsunami, if you like, that in the CAR T cell field, when George Schett and Andreas Mackinson and Erlangen tested CD19 CAR T cells in patients with severe and refractory autoimmune diseases. And this has led those amazing responses observed in a few initial patients have now given rise to a flurry of clinical trials across a number of disorders in both rheumatology and neurology, and I won't say any more about that. Now, tackling solid tumors remains a challenge. And while CAR T cells are being applied with great success, again, in rheumatology or neurology, we're still facing obstacles in the realm of solid tumors. And there are 5 main obstacles, I think. And that's why I insisted on the origins of this field in targeting CD19 in hematological malignancies. The first obstacle is the CAR T cell entry into tumors. Well, we could take that for granted in the hematological malignancies, but that's not the case in solid tumors. Secondly, the immunosuppressive milieu of tumors, of hematological malignancies, is less complex and less potent, if you like, than in solid tumors. And so even if a CAR T cell enters a solid tumor, it may still be inhibited through a number of mechanisms, intratumoral mechanisms. The third challenge is to identify suitable molecular targets. Again, in the case of CD19, we knew that we would induce a B-cell aplasia, but we thought it would be manageable, which it is. And furthermore, it's become today, this side effect is now the therapy in autoimmune diseases, but that's not the case for solid tumors. You cannot target hepatocellular carcinoma and normal hepatocytes. And an enormous effort remains to be conducted to identify suitable targets for this very powerful technology in the realm of solid tumors. And the fourth is the sustaining effective function in CAR T cells. I think most in the field agree that a T cell would have to be functionally persisting longer in the case of solid tumors relative to hematological malignancies. And thus, the engineering strategies need to be further adapted to meet that requirement. And the fifth is that this concerns the delivery of these promising therapies. They have to be effective, of course, but they have to be safe, true. They have to be scalable and affordable. And it's not clear that this would be feasible with autologous manufacturing today. And so there's a fifth challenge here to address. I'm just going to illustrate as quickly as I can the evolution of CARs in 3 studies that I'll summarize very briefly. The first one here brings us back to the very first CAR that combines signaling domains from the CD3 zeta chain and the CD28 chain. We learned from the clinic that these CARs were potent. They are the most potent. They're a little bit more toxic too than the 4-1BB based CARs, and they don't persist very long. And we and probably others thought that initially that this was an intrinsic feature of the costimulatory domain CD28, but eventually revisited that question and introduced mutations in the activation domain of the zeta chain, in fact, toning down the strength of activation. And in doing so, we realized that one particular CAR called 1XX, which retains only one of these 3 green boxes here that are the ITAMs known to initiate T-cell activation, that CARs that retain CD28-based CARs that retained only one of those 3 ITAMs were actually more potent. This did not, the elimination or mutation of the other 2 ITAMs very slightly decreases the potency of these CAR T cells, but greatly enhances their persistence. And this was brought to clinical trials, and we've reported on 28 patients or so now that received these 1XX CARs targeting CD19 and refractory DLBCL, and showed last year that low doses of these 1XX CAR T cells, by low doses, I mean 25 million CAR T cells per patient, were sufficient to introduce very durable responses in DLBCL patients. Second aspect of the evolution of these CARs concerns their sensitivity. It's again the clinic that showed us that some patients relapse not with CD19 or BCMA negative disease, for which obviously you would need to target another antigen, but many relapse with lower levels, lower densities, if you like, of the targeted antigen at the cell surface than present before the CAR T cell therapy. Again, the clinic drew our attention to investigate the sensitivity of these CARs, and that led us to create with Jorge and also Justin, when he was still in the lab, the molecule that's depicted here in the middle called HIT. On the left, you see the T cell receptor, which is well known to be very highly sensitive, embedded within the CD3 complex. On the far right, you see these CARs that I've been discussing so far. And using CRISPR-Cas9 targeting the TRAC locus, we could reconstruct these T cell receptors into these hybrid forms that you see here. And HIT stands for HLA-independent T cell receptor, since the alpha beta chains are substituted with the light and heavy chains of an scFv or monoclonal antibody. And we showed very recently, this was published 2 months ago, that indeed these receptors confer extremely high sensitivity 10 to 50-fold higher than the best of the conventional CARs that we can compare them to, resulting, for example, in the elimination of these renal cell carcinomas or pancreatic or ovarian tumors. You see in the immunohistochemistry an example of these PDX models implanted in mice. You see brown cells that stain positive. Many of the others appear to be negative, but in fact are not negative. They just express very low levels of CD70 and could be very nicely eliminated in these settings using the HIT receptor. And so today we have a diversification of these receptors with different sensitivities. Of course, the affinity and transcriptional control of the CARs make a difference, but their signaling domains definitely do. And you see this hierarchy of sensitivity from the 4-1BB CAR down to the HIT receptor. And lastly, I just want to mention another study recently published for the aficionados here. We recently investigated a component of the pre-T cell receptor, and which is expressed in developing T cells. We had, with Yuge, become intrigued by the vast proliferation of proto-T cells, if you like, that have successfully rearranged their beta chain, which they pair with this pre-TCR, this invariant chain, that is little known in its signaling properties, but is associated with great proliferation of these cells and proliferation without inducing exhaustion. And during this time, these T cells attempt to rearrange their alpha chain until some of them form a productive T cell receptor. And so this led us to investigate this pre-TCR, its signaling domain, which in the lower left you see referred as the 1A domain. And we found that the addition of this 1A domain to the conventional CD28-based CARs considerably increased the functional potency of these T cells, increasing their effector functions and proliferation, but without accelerating their exhaustion. I'll say no more except to say that we found that this is mediated by regulating mRNA translation in T cells. And if you're interested, this was just reported earlier this year in the manuscript mentioned here. So this was just to illustrate that the CAR field is not static at all. Most trials today use one of those two canonical CD28 or 4-1BB based CARs, but there are really newer and better CAR designs to bring to the clinic, especially in the context of solid tumors that could afford greater antigen sensitivity or greater T cell persistence. Before finishing, I want to just turn to the cells that are engineered in these therapies before turning it to Justin in a minute. The initial therapies were, of course, autologous. I say of course because we were concerned that these T cells might be rejected by the host if they were not autologous. And the manufacturing that's been established to date is an ex vivo manufacturing. Initially, it took 10 to 14 days to produce these cells. Most trials in recent times were a 7-day manufacturing. Some of them today are a 3-day manufacturing. And some groups now are even exploring the possibility of an overnight ex vivo manufacturing. A few years ago, great interest arose around the use of off-the-shelf allogeneic cells. Those would be cells that would be made before the patient needed their infusion, ready to go in a cell pharmacy. And there are two main sources for those cells, either healthy donors or pluripotent stem cells. And most recently, what we see is the very exciting topic that Justin will address in a minute that brings us back to an autologous approach, but bypassing the ex vivo manufacturing. Before finishing, though, I just want to emphasize the importance of the progress made in the autologous cell manufacturing to advance all of these possibilities, allogeneic or in vivo. And I want to further address this issue of making better and better cells, which requires, therefore, the infusion of lower and lower doses, or in vivo would require lesser targeting of T cells in vivo. And with Justin, when he was in the lab, we demonstrated that the transcriptional regulation of the CAR is also a very key determinant of the potency of the cells that you will generate, and demonstrated in particular that if you placed expression of the CAR under the control of the endogenous T cell receptor alpha promoter, you would achieve a fairly optimal level of expression of the CAR, not too little and not too much, and that the kinetic response of CAR expression to engagement with antigen was such that it would allow for greater persistence of these T cells. And so we brought this to the clinic at MSK. It's again my colleague Isabelle who designed the IND, and Jay Park who conducted the trials. And I just wanted to mention the first 3 patients that we infused with these TRAC CAR engineered T cells that all receive now 10 million CAR T cells each. And these were patients, again, with relapsed refractory DLBCL. And all 3 went into complete remissions. 2 of them still in complete remission after 27 and 31 months. 1 patient did relapse with a CD19 negative disease. So this just shows to illustrate how improvements in CAR design and improvements in T cell engineering can produce better T cells, which reduces the dose, and I think paves the way for either allogeneic or especially in vivo approaches. And I'll stop here.
Thank you so much, Michel. It was very insightful as always, and you are perfectly in time. So maybe it's the biggest challenge for you working on T cell for more than 40 years, making a long story short, and you nicely show your favorites new CAR T in development. We have a question from Marion Alcantara, Curie Institute, about the binding. Have you ever tested different binder when you use 1XX? Does it make a lot of sense? There is big difference between binders from different scAV, or do you think binding is important, or just the transduction of the signal is most important? Maybe both, or have you ever tested different combinations?
Yeah, no, we've used the 1XX with targeting different antigens, and I believe that Justin will talk about that as well. Of course, the binder is key. The affinity and also the epitope location on the targeted antigen. The T cell receptor can recognize different HLA peptide complexes, but what's remarkable about what evolution designed or selected rather is that these different peptides are always presented at about the same distance from the cell surface in the same molecular context. It's the HLA peptide complex. But CARs, which are guided by antibodies and recognize unprocessed molecules on the cell surface, might bind at different locations on one given molecule. And some epitopes are clearly better than others. I don't think we know many of the rules, except for perhaps proximity to the cell membrane is better. But many of the rules I think, are not understood. And this is often an empirical assessment, comparing different binders. But yes, the affinity is key. The location of the epitope on the molecule are key. The point I wanted to make, though, is beyond that, is that generally speaking, whatever the epitope, the signaling strength makes a big difference in antigen sensitivity.
We have another question about the 1XX. Should we keep, maybe it's a question about deletion or mutation. Should we keep mutated the 2 last Eyquem or just a deletion of, what do you think?
Yeah, this question has been lingering in my lab for years. You can delete the latter 2 items, and that makes a very good CAR as well. But in all our studies, the 1XX is always a little superior, sometimes marginally, not barely statistically significant, but consistently for different antigens and in the hands of different investigators. So I think that the difference is very minor, but very real, which is why we decided to bring 1XX first to the clinic. Why that is the case, we have not investigated in much detail, but I'll just point out that the drawings, as you see them here on this slide, depict the zeta chain as these two vertical rods, but it's not like that at all. This zeta chain is embedded in the cytoplasmic domain, and the regions between the items actually position the zeta chain in a certain way. So we just speculate without any further data that might provide an explanation as to why the 1XX is subtly superior.
Okay, thank you. I have another question for you, and maybe I will just read this question. It's an opinion, so please share your thoughts. Do you think researchers should necessarily go in the direction for solid tumors? Is it a good option?
Oh, I think so. There's an enormous unmet medical need. First of all, we know that T cells are very effective. Checkpoint blockade works by unleashing endogenous T cells. We know that TIL cells work for some patients. So I think those three are absolutely valid reasons for pursuing this space. I'm very confident, actually, that in a few years, maybe not next week, next month, but in a few years, that we will figure out how to effectively target T cells to antigens of our choosing, or probably combinations of antigens, and allow them to overcome many of these suppressive microenvironments. The manufacturing of these cells, which is still an evolving story, as we're going to hear from Justin, I think makes these cells a tool of choice, if you like, when compared to TILs or other strategies for pursuing solid tumors. So yeah, I think it's a very worthwhile effort.
All right, so thank you again. The last question on the Q&A and the chat is about the in vivo. So I think it's a great transition for the next presentation, next speaker. And so again, it's my great pleasure to introduce and to welcome Justin Eyquem. I didn't mention previously, but Justin worked so previously with Michel at SKCC, but for 7 years now, he opened his own lab at University of California, San Francisco. And he make incredible research on this very hot topic about the in vivo CAR T. So thank you again, Justin, and good morning, and thank you for your time. And the floor is yours now.
Thank you very much for the invitation to present some of my work and discuss the topic of ex vivo and in vivo CAR T cells. Before I start, just for those who are not familiar with my lab, I am obviously standing on the shoulders of giants such as Michel. My research program is really based on reprogramming, engineering immune cells, in particular T cells, but we also have other research programs in NK cells, B cells, and also hematopoietic stem cells. The way that we're thinking about engineering cells, and again, it's very inspired by Michel's approach, is on one end, systematically engineering proteins, and not only the receptors like CAR or TCRs, but we now have a deep interest in tuning and reprogramming T cells with novel signaling proteins, whether they are variants or fusions. We've been using a lot of genome editing, and I'm going to talk about it today, not only to manufacture CAR T cells, but also as a tool for functional genomics and identify gene circuits that could be associated with CAR T cell functions and dysfunctions. And finally, as we established my lab, and more by necessity really than training, we got really interested in delivery, because at the end of the day, if you have a great CAR and a good way to place it precisely within the genome of a T cells, you still need to have good delivery methods to facilitate the manufacturing, both ex vivo and more recently in vivo. And so just to touch base, but Michel already introduced targeting a CAR into the track locus. It really stems from these observations, which is well known now for decades, that the gamma-Y12 vectors or lentiviral vectors that have been used in all of the FDA-approved products integrate in a semi-random fashion throughout the whole genomes and tend to integrate close to transcriptally active genes. And now there's been multiple reports of transgenes integrated in coding sequence or introns of coding sequence disrupting those genes. And one example reported at UPenn and showed that here a CAR transgene introduced into the introns of the tumor suppressor TET2 genes and associated in these specific patients with a second allele that was also mutated, provided a full knockout of that tumor suppressor genes and was associated with a massive clonal expansions. And at some point, 100% of the circulating CD8 positive CAR T cells were all stemming from a single clone. And I think here, in this context, this clone actually provided the therapeutic activity and did not turn into a secondary malignancies. There now has been multiple reports showing that this can happen, including one reported by Michel, where the CAR transgene integrated into the TP53 genes. And here in that context, I think it shows an exciting engineering challenge, showing that potentially if you engineer one cell really well, that might be enough to provide a therapeutic activity in patients. Now the real question is can you do that in a really safe and efficient way? And so another potential issues with those viral vectors is that they always, in all the FDA-approved products, use constitutive and very strong promoter, which means that any cells that are transduced with the vector will express potentially the CAR. And if it happens in a tumor, the CAR can eventually interact with the antigen it's trying to target and hide it from the cell surface, potentially creating a tumor, an antigen-negative tumor escape. And this was reported with ex vivo manufacturing at UPenn. But now that the field is moving from ex vivo to in vivo manufacturing, and we don't have the ability to do all the quality control, the purifications, the selections of the T cells, we need to make sure that those vectors are not transducing any tumor cells in vivo. And so, as Michel introduced, while I was in his laboratory, we thought that we can move away from those randomly integrating vectors and place it into one specific locations. And while we tested multiple loci in T cells, we found that the track locus was optimal. And we optimized the first version of the protocol using electrophorations of CRISPR RNP targeting the first exon of the track and an AAV6 to deliver a donor template in which we're using a promoterless cassette, meaning that the CAR will not be expressed except if it's integrated into the track locus. And if this is in the T cells, because the track locus is a lineage-specific promoter that is exclusively expressed in T cells. So now it's providing a lineage-specific expressions, which we found is really important, especially as we'll be moving to in vivo. And so one advantage of this technology is that it offers a very homogeneous and predictable CAR expressions. As you can see here on the right, in blue, all of the different donors that were tested, and now we probably tested hundreds of donors, always have exactly the same level of expressions. This is very homogeneous and predictable, so you really standardize the way you're manufacturing T cells, and everything that you're establishing in your lab, the functionality of the CAR, the functionality of the payloads that you might deliver next to the CAR will always have the same expression level. And now we know by experience that the expression level matters a lot for the functionality of both the CARs and payloads. And so now having a tools to make sure that whether it's in the lab or in patients, we always have the same expressions actually save us a lot of time. Also, as Michel introduced, we found that in this first BLL xenograft model, that a single injections of the track CARs was outperforming CAR manufacturers with gamma-Y12 vectors in this experiment, which was a stress test in which we were injecting very low dose of CAR T cells. Looking into the bone marrow of those mice where both the tumors and the T cells are homing, we found that the tracked CAR T cells had a phenotype associated with a more functional phenotype than those engineered with gamma-12 vectors. So now we found that this strategy has been safe because it's targeted, it's promoterless. It also provides homogeneous and predictable expressions. Michel introduced the transcriptional regulations that the track promoter offer, which we found is what really improved the functionality of CAR T cells, has a very specific expression level that can always come back to baseline and also provide a break in T cell during rounds of killing. We also find that this platform is very flexible. We can introduce many types of genes, and we can also use that for multiplex editing. It is scalable, as Michel introduced. It can be in clinical trials, and it's also adaptable to every editing platform. And what is interesting is that now this is the most targeted locus in T cells. There has been more than hundreds different ways that the field has been now targeting this locus, and pretty much every time a new genome editing technology is being introduced and people want to test it in T cells, this is the first locus that they're going after. The technology has been licensed to many companies and now has been used in more than 10 clinical trials, including 2 academic trials, one that Michel introduced at MSKCC, and a second one that is now just opening at UCSF, for which Alexis, who did the introductions, designed the CAR for, and this is for multiple myeloma. But now the topic of the day is really in vivo CAR T cells. And so how can we bring all of this technology and all of the precisions that we brought to the field from ex vivo to in vivo manufacturing? And so the goal of in vivo manufacturing is really, and the challenges are, can we engineer enough cells in vivo? And can we do that in a precise enough way that it maintains the safety of the patients? And so the current methods that are being explored all rely on using vectors that can deliver the CAR genes either in an integrative or in a transient way into T cells. And so 2 main technologies are currently explored, both preclinically and clinically. The first one is really based on engineered lentiviral vectors. Those are pretty alike the lentiviral vectors that are used in FDA-approved products. The main difference is that there's been some further engineering of the envelope to provide more specificity in the delivery in vivo. And this is way in most products in the following passions. Lentiviral vectors to transduce a cells needs to both have a binding of a receptor on the cells and then a fusogen activity. And in most vectors that were being used, both are offered by the VSVG glycoproteins. And it has been found that some selective mutations in VSVG can maintain the fusogen activity while removing the ability to bind to its cognate receptor, the LDL receptor. And in that way, if you're pairing that mutated VSVG with an antibody, you can specifically transduce the cells that are expressing the antigen specific for the antibody. And so if you want to target T cells, now you can use an antibody for CD3, CD5, CD7, and others. Now, this technology, the only specificity that is provided now is through this envelope engineering, because most products are using a constitutive promoter. It still provides these random integrations that can be associated in some rare cases with tumor suppressor inactivations, which still bring some risk. And those constitutive promoters, if this first line of safety and specificity offered by the envelope fails, it means that any cells that would be transduced would express the CAR. And if it happens on the tumor cells, as I mentioned earlier, it might be an issue as it can potentially hide the antigen from the cell surface. And so the other potential issues with those vectors is that because they're using constitutive promoters, in the producer cells that are used to make those vectors, the CAR is actually already expressed and already displayed on the producer cell surface. And when the virus is budding out of the HEC cells, it can be actually displayed on the lentiviral particles, now providing tropism toward the cells that they're actually targeting. And so this, and it's been also reproduced in my lab, is a potential risk, especially with the use of constitutive promoters. Now, the other technology that is being currently tested is based on transient delivery of mRNA using lipid nanoparticles that can be targeted also with antibodies to provide more specificity toward T cells. So while the targeting can provide more uptake by the targeting cells, it is understood that lipid nanoparticles do not provide the same specificity of delivery than the virus particles. And so we have to know that many other cells in the body can be transduced with these LNPs, even if they are targeted. Now, most technologies are using mRNA delivery, which only going to be expressed transiently, and so does not bear the same risk of randomly integrating transgenes or stably expressing a CAR into our tumor cells. Both technologies are currently tested clinically, and with some very exciting preliminary data suggesting that it is possible without lymphodepletions to achieve very good response, and in some cases, complete responses been observed in multiple myeloma and currently tested also in B-cell malignancies. So this is really exciting, but I think this is the very beginning, and we're already thinking about how can we improve this technology and provide more safety and efficiency to the platform. And so when my lab started being interested in developing in vivo technologies, we decided that we did not want to lose the specificity and the standardizations that we spent the last decades optimizing. And so we wanted to maintain the precisions by moving in vivo. And so some advantage of doing site-specific engineering in vivo, it will maintain the precisions of delivery. And here also, as I explained earlier, we will be delivering a promoter-less cassette, which again would not be expressed at any moment during the productions of the vectors and can only be expressed once integrated into the track locus and if it is happening into a T cells. So now it's providing really an expression specificity that the other platforms cannot really offer. Now, the issue when we started this project is that achieving homologous directed repair in vivo in primary immune cells has still have not been performed. And the main challenge was to deliver enough DNA of the donor template within the nucleus of the targeted cells, the T cells, without having too much toxicity. And so here we need to deliver two reagents. One is the Cas9 to have the cut and the double-strand break. And as I mentioned, the other one is the donor templates. For the Cas9, we really want to have as much specificity and as short of expression as possible to both minimize the immunogenicity and the off-target. And so one part of the equations was actually already solved by the Dana lab, who developed some virus-like particles. Also, they call it EDV for enveloped delivery vehicle. That from the outside really much looks like lentiviral particles, but in the inside does not deliver any nucleic acids, but instead deliver a Cas9 RNP that is fully formed and ready to cut and fuse to the GAG polyproteins and can be delivered to the target cells after transductions. And the same tricks of envelope engineering can be performed with those particles. For the donor templates, we know that adeno-associated viruses can naturally evolve to deliver single-stranded DNA into the nucleus of its target cells. in a very efficient way without raising too many alarms. And we know that AAV6 can transduce hematopoietic stem cells and lineage deriving from it, such as T cells. And so we started with potentially AAV6. One advantage also of single-stranded DNA is that it works remarkably well for homologous directed repair, which is not the case, by the way, if you're using recombinase or transposase, which are other technologies that are currently explored. So now for the EDV, we used a CD3 targeting EDV that was developed by Jenny Hamilton in her Nature Biotech paper, in which a mutated VSVG is paired with a CD3 antibody that can simultaneously provide specificity towards T cells and also activate T cells at the same time. For the AAV, we used an AAV capsid evolutions, which we did in collaboration with Aravind Azokan. We previously reported similar evolutions for mouse T cells, but here we did it for human T cells. And the goal was to both create an AAV that can be resistant to pre-existing neutralizing antibodies and to limit the tropisms of the AAV towards T cells and less of the other cells that AAV6 can transduce. And so here it's a library of about 1 billion AAVs with randomized 7-mer amino acids, which were selected because we know they were important for primary receptor interactions and interaction with neutralizing antibodies. And after 3 rounds of evolutions in the presence of human serum, we identified a novel family of AAV, including one which we call AAV-HT7. That AAV, here ex vivo, maintains the ability to achieve efficient knock-in at the track locus, even in the presence of human serum. While AAV6 here, as soon as you include serum and you lower the MOI, is directly affected by the neutralizing antibody presence in the serum. So to test the specificity of our vectors, and sorry, here ARC312 was the previous name of this AAV, but now it's also HT7. We use a different locus, the CLTA locus, that is expressed in every cell, and we're delivering a GFP right after the start codon. So any cell that is transduced turns green. And so here what we see is that with the wild-type VSVG and AAV6, you are able with that dual vector to transduce and achieve knock-in in T cells, but also in NK cells, in HSCs, in macrophages, in a bunch of B cell lines, suggesting there was no specificity at the delivery and engineering. Now, when we're moving to our engineered vectors, the CD3 EDV plus the AAV-HT7, you see that exclusively T cells now are being engineered and all of the other cell types are clear of engineering, suggesting that now we have specificity at the delivery, engineering, and expressions offered by the track locus. So now, testing those vectors in vivo, we use an NSG that has an MHC class 1-2 double knockout to limit the xeno-GVHD, which we know would pre-activate the T cells and make it easier to engineer them. So here there is no much GVHD. We are delivering a CD19 1XX CAR that Michel introduced earlier, which is helpful here because we can both look at T cell generations and B cell aplasia as a surrogate for the activity. A single injections of the 2 vectors found that in the conditions with our best 2 vectors achieve up to 15% of those planocyte T cells 2 weeks after single injections. And this is the results of both engineering and clonal expansions of the CAR T cells that were generated. In the best conditions, we were able to achieve complete B cell aplasia, which is relevant to the autoimmune disorder field that Michel introduced also earlier. But now we're interested in oncology. And so we first tested it in the NAM6 models, which we know is a very aggressive B cell malignancies, and tested after a single injections and found that it was enough to mount an anti-tumor response to achieve complete response in most of the mice and one partial response. To test if we achieve the functional persistence that is associated with the track CD19 1XX when we do it ex vivo, we re-challenged the mice and found we were able to control a re-challenge of a high dose of NAM6 40 days after the first injections. Now, how does it compare to other delivery methods, such as in vivo lentiviral particles, which are the ones that are, for example, developed by Kelonia or Interius, and others. So we devised a large experiments where using the same donors, we tested both ex vivo and in vivo comparisons of T cells generated with lentiviral vectors or track targeting, and all of them delivering the same CARs. And for in vivo, the lentiviral vectors were displayed with the same antibody than the EDV. We found that in vivo, at similar dose, site-specific engineering at the track locus was yielding a much stronger anti-tumor response, achieving complete response in every single mouse here. Now for the lenti, and we used two different dose equivalents to be as fair as possible. The first one is based on transduction units, which is the level of engineering you get ex vivo and what dose is necessary to achieve equivalent. And the other one based on particle numbers, P24, which give us an ability to inject the exact same number of particles than EDVs, and is 3 times transduction units. With both equivalent, we found that we have lower response than with site-specific, suggesting that at similar dose, we get a stronger response with TRICAR T cells. So why is that? Now in the paper, there's much more data, but just highlighting one aspect which we believe is really important. If you look here in blue, those are cells that we found in the spleen and in the bone marrow. You see that whether they are engineered ex vivo or in vivo, with site-specific engineering, you always have a functional level of expressions that is very homogeneous, while with ex vivo lentiviral vectors or in vivo lentiviral vectors, you have a really heterogeneous, variegated, and sometimes very low expression, especially with the in vivo lenti. To zoom in a little bit and compare site-specific versus in vivo lenti, with site-specific, you have actually exactly the same level of expressions that you have ex vivo, meaning that everything that we've been optimized for the last 10 years at the track locus ex vivo will translate identically in vivo, which is going to save a lot of time for us in terms of optimizing the CAR and the payloads. With lenti, we only found this little shoulder of expressions. And for those who've been using lentiviral vectors in the field for a long time ex vivo, we all know that the level of expressions and the transduction rates that you obtain with the lentiviral vectors is directly linked to the MOI. How much virus and what is the concentration of the virus that you use. The higher the volumes, the more expressions, and as you reduce it and you reduce the MOI, you just obtain this little shoulder. Now, when you're injecting the lentiviral vectors in your blood, what you're pretty much doing is diluting this MOI down to infinite. And so having a low expression level on average like that makes sense. And so what we believe is that with site-specific engineering, every cell that you engineer is going to have a functional level of expressions, while with lentiviral vectors, only a subset of those cells will have a functional level of expressions, which will eventually expand and achieve potentially some responses. But at equivalent dose, you get a better response with site-specific. And so now we believe that by combining the cell specificity of multiple vectors and lineage-specific endogenous promoters, we have 3 levels of specificity, which really contains the expressions of the CAR to T cells. And we are able to achieve B cell reset and complete response in multiple tumor models. I did not have time to show it here, but we're also able to achieve this type of response in myeloma and in sarcoma. And now we're also already incorporating the large synthetic immunology toolbox that collectively the field is being generating and putting it into our system. And we think that in principle, this is applicable to many other cell types and other therapeutic use. And so I'm going to stop here and thank my whole team and thank you for staying online.
Thank you. Thank you so much, Justin. It's always fascinating what you can do now and impressive data.
Thank you.
We have a question about the safety in the chat. So do you think, Justin, there is a higher risk to develop a T-cell malignancy when a T-cell knockout is induced in the majority of peripheral T-cell in vivo?
So here, when we do in vivo engineering, you have to see that this is what we call a light touch engineering. it's actually very few T cells that are losing their TCRs. And if they don't have a TCR and they don't have a CARs, they do not expand, and they tend to actually die. And we don't find much TCR negative T cells. Um, and so we also have been developing other strategies, which we reported last year in Nature Biotech, that show that you can target introns, which is what we're doing now. And so the advantage of targeting an intron is that even if you have indels, you will not knock out the TCR. And it's only when you're knocking in the CAR that you lose the TCR. And so we found that this is probably a safer way to limit knocking out TCRs in more T cells in the body.
Is it possible for you to share? I think you have some preclinical experiment or some data about what will be the future for the in vivo from what you already exploring. What you can say.
I mean, I think the future comes with precisions and comes with more payloads to be tested, especially for to expand it to other diseases than hematological malignancies. Michel highlighted the challenges that are associated with those. And I think the good news is that we have, as I mentioned, a really large toolbox already. Now the question is like, how fast can we test those tools in the clinics to see if they can be efficient and safe? And those can be already done ex vivo and then ported into in vivo. But I think the greater challenge for the in vivo, and this is very early and we don't have the data yet, is how much persistence we're getting from those earlier very exciting responses, and what is the safety profiles of the different vectors that we're using. Because now we not only have to deal with the potential toxicity associated with CAR activity and expansions or of targets, but also the potential toxicity associated with the vectors that we're using. And so understanding those responses is also going to be important.
Justin, one question. Do you think it's a good approach to maybe deliver directly both particles intranodally or directly into the spleen, so you increase the chance to have one T cell getting both particles? And then do you know, it's very theoretic, but how many cells are in the end getting both particles that could explain all this final expansion? Do you have like, you cannot
Yeah.
follow these cells because they don't have a TCR anymore, so it's hard to follow clones, right?
I mean, we can, at the transcriptional level, we can still, we do TCR sequencing, and so we were able to have the clonality. What we found in our mice is that we found about hundreds to thousands of TCRs, and so that's the minimum number, but we know that clonal expansions is not homogeneous. And even when you were injecting hundreds of million CAR Ts into a patient, if you do TCR sequencing, you actually find a subset of the cells actually expanded. So now we know that at least hundreds or thousands of cells were engineered. Now, in terms of each vectors has a very different biodistributions. AAVs actually diffuse remarkably well. And we found, and in the paper we reported last year for our mouse T cells AAV, we found that 20% of every cells throughout the whole mouse, and we looked in pretty much every tissue, about 20% of those were all transduced. And so it's a really large number of cells. What is limiting really is more the virus-like particles that dictates the number of cells that will be engineered. So it's not the intersections of 1%. It's more one is very efficient and the other one slightly less. And so that's really the number of cells that are transduced by the VLP that dictates the dose. Now, what we know about clonal expansions from ex vivo CAR T is different in vivo CAR T, and we think that not going through the exo manufacturing maintains a lot of their clonal expansion capacity beyond what has been seen with ex vivo CAR T. And so, especially for hematological malignancies, I think that the number of cells that will be needed to be engineered in vivo is fairly low. And I think what Michel saw was that we can get complete responses with 10 million CAR T that are well manufactured and delivered a really good CAR. I think it tells you that, and this is after a week of manufacturing and freeze and thaw. So It's actually probably from the beginning of the manufacturing, few thousands of cells, that can do the job. And so I'm confident that we don't need to maximize the number of engineering, but carefully craft a good car and a good way to deliver it.
Sure. There are some questions in the chat.
I can do a short question for you, Justin. Is a dual targeting approach with in vivo feasible? I mean,
Yeah, we already have data on that. And yeah, we can deliver two cars. We can deliver bi-specific cars. Both are working really well. We now been able to do an experiment where we're delivering 4 genes into the track locus in vivo, and the 4 of them are functional. And so as long as it fits into the AAV, which is about 4.7 kb, you still have a lot of room to play, and we found that it's been working well. Yeah.
Cool. So your first trials will necessarily be in last line patients, I guess, even maybe eventually after CAR T cell treatment. Do you think, I mean, the T cell fitness and that you will find there, I mean, you'll have probably some cells that are very dysfunctional. Is there a way to somehow try to target through AVV evolution more like memory cells, or do you think this could be an approach? And so what's the path to go eventually to earlier lines of therapy? First, you need to prove, of course, efficacy, but what are your thoughts about all
Yeah,
that?
no, I mean, I agree that for first in human, we're always going to have a populations of patients that have going to have been heavily pretreated. Now, a lot of companies are actually exploring first in human outside of United States, where fewer of those patients have been pretreated with CAR T cells, and so that is something to keep in mind. Now, I think some of the earlier patients treated with in vivo CAR Ts were also heavily pretreated, and I think they showed that they could still have some good responses. Now, it is possible to engineer your vectors to be more selective into the cells that you're engineering. But I would say that this is still a numbers game. And so now if you're restricting the populations that you're targeting, the chance to have enough CAR T is also a challenge. And so there are definitely some elegant and specific ways to achieve that. But you just have to know that now you have fewer cells that you can engineer, so it comes with other challenges. Now I think that the car design, and I think Michel explained this really well, plays a huge role in the way those CAR T cells are functioning. And I think crafting an optimal CAR design for those indications is really important, and at least as important as selecting the right cell population.
Is there maybe an approach to somehow adding like a bispecific molecule to approach this two particles, to T cells or to some specific T cells? Do you think this is something feasible? Or...
Yeah, no, you can definitely use other antibodies that will be targeting, any subset of interest. So it is possible. And also it goes in both directions, because when the particle transduce cells, it actually deliver signals to cells. And so here we know that the CD3 targeting that we're providing does activate the T cells. And so you can also use the particle to display molecules and ligands that can provide co-stimulations or activation of T cells in a way that would benefit their function. So it's another axis that we can use.
It's almost impossible for me to stop you guys, but we are reaching the end of this discussion and seminar. So maybe, Jaime, you want to conclude?
Sure. Unfortunately, it's impossible to continue the discussion, although I'm sure that everyone would like to continue. But thank you, Michel and Justin, again for such rich presentations and discussions. And thank you all for your engagement throughout this RedTalks season. And please take a moment to fill in the satisfaction survey at the end. And stay tuned for season 6, episode 4, which will explore bispecific antibodies and versus CAR T cells. So another very exciting topic. So thank you again, everyone. Thank you, Michel. Thank you, Justin. And thank you, everyone, for your questions.
Thank you.
Have a good evening and good day in the United States.